This is a podcast of the Nuffield Department of Medicine. Today we talk to Dr. Alex Bullock about understanding growth signals. Hi, Alex.
Hi.
Why is it important to understand growth signals in cells?
Growth signals are fundamental to many disease processes like cancer. Growth factors have to be carefully controlled so that our body develops in a unique pattern, and too much growth factor signaling can cause, for example, the development of tumors. It can also cause the development of many developmental disease. Perhaps the best known growth hormone is human growth hormone, which is used by athletes to sort of gain competitive advantage. But too much hormones and too much growth factors is not always a good thing. We're particularly interested in bone morphogenetic protein hormones that control bone development and stem cell differentiation. Now, we're very interested in one particular disease called FOP, or FIBRODYSPLASIA ossificans progressiva, which results from too much signaling in that pathway. And that causes the breakdown of muscles and infiltration of stem cells, and then the differentiation of these stem cells into bone. So what happens is that patients from early childhood get slowly paralyzed by bone formation in their muscles, and they lose most of their movement. So it's a very debilitating and sort of paralysis-causing disease, and we're trying to understand both the causes of the disease and develop new treatments to counter that disease.
How does your group actually look at this kind of problem?
We take a multidisciplinary approach, but one of the major focuses is to learn more about the 3D structures of the proteins, the molecular machines in our body that are kind of doing the work for us. And we use the structures to identify various properties about the protein. So we would like to learn, for example, how disease modifies these proteins, how mutations in the proteins change their structure and activity. And we also like to use whatever activity assays we can, so we can kind of build a correlation between the structure of the protein and its function. And so we can look at binding assays for various partners that cooperate and work with the protein and signaling, and also the interaction of drug molecules that might be useful in fighting disease.
What kind of techniques do you use?
3D structures of proteins. The structures of proteins are not visible by the naked eye, so we have to kind of use another technique, and the technique we use is called X-ray crystallography. Basically, this involves shining really powerful X-rays, protein crystals, and just from the way that the X-rays scatter off the protein, with sort of clever mathematical modeling, we can back calculate what the original structure of the protein was that sort of scattered the X-rays. On top of that, we use a barrage of techniques to delve into the protein activity in more detail. So the structure really just gives us a static picture, and what we want to do is know a bit more about its function and activity. So for that, we can use a range of binding assays like fluorescence, binding assays where the signal changes upon binding, or we can use luminescence, and we can also use those techniques for our drug screening assays as well.
What are the most important lines of research that have emerged over the, say, last 5 to 10 years?
I mean, most fundamental to us has been the Human Genome Project and the long-awaited era of genomic medicine. The human genome is really the code book for life, and that's our tool for understanding all the disease processes and normal development. And what we would like to do is to take what we do on a daily basis, if you like, is to read parts of that code and then try and interpret them in terms of understanding what is that code doing, how has it gone wrong. So for example, in the FOP disease that which we study, we know that there's a single point mutation, just one letter in the DNA code is changed, and that's sufficient to cause unwanted bone formation in muscle. So now we know that we have that point mutation. We can then say, what is that doing to the protein involved in FOP? How's it changed its activity? And can we find drug molecules that can block that change in activity?
Why does your line of work matter? Why should we put money into it?
Well, FOP is just one of 7,000 rare diseases. And these affect almost 10% of the population in total, even though individually they're rare diseases. So there's a great unmet medical need. Possibly only just a handful of these diseases have at the moment any form of medicine that can help mitigate the symptoms. So there's a huge medical need to counter that. So what we've done for FOP, we would like to translate into other diseases. And we have a 5-year program funded by the Wellcome Trust to kind of support early investigations that could feed into drug discovery programs, particularly in cancers, metabolic diseases, and sort of neuropsychiatric diseases.
So how then does your work fit into translational medicine within the department?
Our most advanced study has been on FOP. And for that, we've identified an investigational drug that's currently been treated in several thousand humans. And we would like to take that drug, which has mainly been trialed for cancer, and repurpose it into a clinical trial study for FOP. And ultimately, after that, we would like to take that approach, if you like, of what we've learned about developing small molecules for FOP and try and expand that to other related conditions to FOP. So what we would like to do is take basic research, understand the mechanisms and the sort of drug binding characteristics of protein syntheses, and then translate that into drug discovery programs across the country and also into clinical trials.
Thank you, Alex. That
Thank
was
you.
really interesting. Thank you.